think that it will be very exciting and educational for all of us to be able to, pursue these types of global discussions amongst the entire staff to increase our understanding and our ability to be able to explain things to patients. So what I want to do today is to present a little bit of background on what's called systems analysis. In other words, interrogating all the omic data, the genome, the interactome, the proteome, all these different levels of data that we're now collecting as we move from gene to mRNA to protein, and just give you a general idea of what's going to be necessary to take all this data that we're now beginning to accumulate on patients and to put it together to allow us to be able to actually personalize our therapy. And as you can see on this first slide, it's clear that as you were able to see on the first slide before it was changed, you essentially can see that in order to really understand how we can regulate genotype, the basic template, there seems to be a lot of noise on the line.
Yeah.
Okay.
Stop muting, guys.
Use your line, please. I'll bet Dr. Senzer.
Yeah.
So in order to understand how we can regulate the template that is on the gene and be able to use the information on the gene and what is encoded via transcription and translation, how basically we have to be able to put that together to approach our patients individually. And then once I just give you this little bit of a general introduction in a few slides, we'll move on to something that we just recently looked at to show with the data we currently have how we basically can indeed come up with some ideas about how to address the specifics of transferring this data to personalized therapy. So now slide number 2. So the gene regulatory network actually, as I already suggested, is multi-layered. On the bottom, we start out with the genomic level. In other words, what's actually encoded on the gene. Now keep in mind, as we talk about the genomic level, and I'll be showing a slide a little bit later to address this, there are really 2 parts to this. One, of course, is what is structurally present in the gene, the mutations, the fusions, the insertions, the deletions, all those structural changes that we are currently looking at and getting feedback from a number of the different agencies and facilities that we're working with. But then there's also a second type of information called the epigenome, and those are basically gene-related changes that are heritable, that can be passed along, but that are not structural, and that has to do with a group of islands we call CPG islands, cytosine and guanine, and whether they're methylated or demethylated, which turn genes on and off. So we have that one level, which is the genomic level. Then, as I think we probably all know, the gene, then the information is transcribed into messenger RNA, which forms a second level, which is called the transcriptome, which isn't on this slide. And then the transcriptome is basically put on yet a third level, which are the proteins, which are really the workhorse, and are those entities, those molecules, which actually carry out the functions encoded by the gene. Now, one of the problems we've had with all the excitement about being able to deconstruct the genome and take all the mutations and the different structural changes that are present in our patients and be able to figure out what to do about it, is that basically just gives you what the cell, what the organism has a potential to do. It doesn't tell you what it's actually doing. So in order to know what's actually happening, you really have to look at all these levels. And looking at this slide gives you some idea of the problems we face when we just look at that lower gene level. And, you know, you have the genes, and as I said, they transcribe mRNA, and then the mRNA is translated into proteins. Well, we also know that once you have those proteins basically can form transcription factors. And those transcription factors mean that the proteins themselves can feed back onto the gene level. And by modifying the regulatory units that turn genes on and off, the proteins can actually control the activity of the genes. So we really have to know all these levels to be able to understand not only what the cell, particularly the cancer cell, is capable of doing, but to really understand what it's actually doing. Next slide. So this basically is what our goal is at the Crowley, to be able to really get a molecular fingerprint on each one of our patients. You know, we start out with the tumor and the normal cells from the patient so we can see how the tumor is different from the patient's normal cells. And what we want to do is we want to, you know, really break down the tumor genome. We want to know what are the structural and non-structural changes in the tumor gene that differentiate it from the normal cell, and that includes not only mutations and all the different structural changes, but it also includes the numerical changes, what some people have called the amplicone, because we do know that in some tumors we have multiple copies of a gene, and that gene is what we call amplified. And when you have an amplified gene, you have a much greater likelihood that that is going to be transcribed and translated into a higher protein expression. If you basically have, you know, a decrease in gene number, the reverse could happen. So we need to know all this data, couple that with the transcriptome, in other words, the RNA, the messenger RNA readouts, and then we want to know how that is translated into proteins. And ideally, not only do we want to know the proteins and what proteins are being expressed, and probably more importantly, what proteins are being overexpressed in comparison with normal tissue, but we would also like to look at what is called the kinome, which basically tells us which of those proteins which are being expressed are active at any given moment. You can have proteins that are expressed but are not necessarily active in a process. And the kinome generally includes which of the proteins are usually, in terms of activating them, phosphorylated. And that way we not only have an overall view of the proteins that are expressed and overexpressed by the tumor, but which ones at a given time are active in the cancer process. So we want to identify as much of this data set as possible on the individual patient, and then we still need to put it into context. And what we would like to be able to do is to take all of the human data in the literature, you know, not the mouse data, not the culture data, but the human data, as well as the data we at the Crowley are accruing from our expanding surgical base, and then use both of those sets of data, the patient data and the generic data plus our own subset of data, in order to try to better understand these interactions between these multiple levels. We want to then take all this data and integrate it, put it together in both a horizontal and a vertical fashion, meaning compare patient genome to our database genome and patient's transcriptome to our database transcriptome, but we also want to do this vertically, meaning comparing the genome to the proteome to the kinome on both the patient level and our database level, and be able to integrate that, come up with an algorithm in order to be able to determine which of these sets of genes and their products seem to be the most pertinent for that particular tumor, and then a method of scoring them so we can prioritize these different potential targets. And then once we've done all that, pick out the targets that we say, okay, these are the targets that we think we need to attack to essentially break down this tumor, hopefully get a response and benefit our patients. So next slide. So what are we looking for when we say, okay, we want to get a target? What is the ideal target that would allow us to effectively attack the cancer? Well, one is that we need to find a target that's crucial to the malignant process. And most of us now distinguish between what we call driver and non-driver genes. In other words, genes that basically have a controlling effect on the either the onset or the continuation of the cancer, and those mutations that just happen to occur, and even though they're there, may have some subciliary role, but don't really have a major controlled mechanism for the tumor. So crucial to the malignant phenotype. The second characteristic is that it really shouldn't be, it should not, definitely not be expressed in vital organs or tissues, because if we start targeting, a gene that is expressed and important in the ongoing maintenance, obviously, of the rest of our body, that significantly increases the risk of side effects and limiting the use of that therapeutic. It has to be allow us to be able to say that it's biologically relevant, especially with regard to the biologic relevance of the models we use. So one of the things that we discuss when we are planning within Gridallis to come up with, do we want to use a hamster, a mouse, a monkey, in order to be able to see not only if a therapeutic is effective against the cancer, we have to find a model that's relevant, that allows us also to be able to see whether or not it can have potential side effects in humans. And that actually makes us compare the genes of these different models to the human genes to be able to make sure that we are getting the best of the preclinical data in choosing our ideal target. The target we choose has to be reproducibly measurable in clinical samples. In other words, it doesn't help us if only 1 out of 1,000, has a particular, targetable process. It can help that patient, but it then becomes extremely hard to get funding from a cost-effective basis in the current, healthcare cost environment. The target we choose has to be correlated with clinical outcome. And, certainly we need to have a target where when we knock it out, when we try to negate its effect, we can get a clinical response, a significant proportion of patients who actually do have that target. And the converse would be that if we have a group of patients that don't have the target, and we use a therapeutic, we don't really expect the response. So these are the characteristics of the target that would enable us to feel comfortable that if we come up or are authored a therapeutic that has all these elements, that it will have a high likelihood of effectiveness. Next slide. Now, I mentioned to you driver genes, and just a quick review for everybody, who, hasn't, really gone into too much detail on this. There are 3 main types of driver genes. These are those genes that are important for the malignant process. And one group is called the oncogene. And oncogenes can activate cancer activity and allow for continuation of cancer activity. The second group of genes are called tumor suppressor genes, and these driver genes essentially decrease the ability to block abnormal cell proliferation. So oncogenes are important if they're present. If we say something is an oncogene, we are basically saying that when it's present, it causes a problem. On the other hand, the tumor suppressor gene, since it normally inhibits abnormal cell growth, if it's absent, it's a problem. Now, that we can't target, obviously, because the problem is that it's absent. That becomes a driver gene that we need to replace. And then the third type of driver gene is called the stability gene. And that's become more important recently with the therapeutics that are becoming available to affect repair response, because stability genes are those genes that affect the cell's ability to repair damage. And one of the problems, of course, with cancer is that when you have changes, genetic changes, that impair the ability to repair damage, you wind up accumulating more mutations. And by accumulating more mutations, you significantly increase the risk that you will get a combination of these mutations and abnormalities that basically allow for the development and then maintenance of the cancer. Now, when we take all these driver genes, these three types, we then have to say that there are two roles for the driver genes, which I've kind of already touched on. First, there's a causal role, an initiating mutation of a gene, or more likely with solid tumors, a combination of genes that leads to the development of the malignancy initially. And then there are driver genes that may not have been the initial changes that led to the development of the cancer, but are absolutely indispensable for that cancer to maintain itself in an active fashion. So those are the two roles of the three categories of driver genes that we're looking at, well, I'm looking for. Next slide. Of the ways in which we try to determine, of the different target genes that come up, I had mentioned to you that not only do we have to integrate the data and develop algorithms, but we have to prioritize and score them. What are potentially the most effective targets for our therapy? And a concept has come up called betweenness. And, you have a network. And look at all these different dots, the gray dots, the blue dots, the green dots. Assume that these are all genes for the purposes of our discussion. Essentially, betweenness means that if we have a node or a gene, one of these circles, basically, when we score them, let's look at the different pathways connecting these different entities, going through any one of them, and try to pick out the ones that seem to have the greatest number of the shortest non-repetitive paths going through them. The reason this becomes important is if you look at this diagram, especially this intermodular hub, you see that on this particular slide, there are like two big modules, one on the left and one on the right. And this intermodular hub is obviously a big information transfer between these two modules. And if you knock out these intermodular hubs, these intermodular driving genes, you basically split the crosstalk between these different important complexes apart and have a higher probability that you're going to affect the cancer in a significant way than if you knock out, let's say, that orange hub, which, will cause, some disarray, but it's not going to really significantly affect the massive amount of information flow that any cell or process needs in order to continue being active. So this concept of betweenness and the ability to be able to take that concept and using that pick out these key informational transfer points between genes, which we call bottlenecks, gives us a much higher probability of finding a target that will be a major player in the cancer process. Next slide. And on this next slide, I kind of picture this for you. If you look at, looking at these two, different bottlenecks, the major one that blocks the most amount of information transfer versus the hub that essentially will cause some decrease in information, but not in a major way. And you look over at the right and how much it disrupts the processes, the kind of reddish tinged box. If we set that as 1, for just, randomly knocking out a gene, and, the level of disruption it causes, if that's a 1, and we then say, okay, let's take a look at some of these intramodular hubs. Those are the ones I showed you, that little circle on the previous slide, which has some effect, but not a major effect on information. Generally, that type of mutation is more responsible for homogeneity, holding processes in a homogeneous or a similar process. If you knock that out, you do actually have some degree of disruption. In this yeast model, actually, it was about a 30% increase in disruption. But if you knock out those major interference information flow hubs, those intermodular hubs, as you can see, that has a 300% increase in disruption. And the best way to visualize this is on the next slide, where I basically show you up above a network where you've kind of had some random hits on it, and it still pretty much functions as a network. Then I basically show you on the bottom, when you kind of get the intramodular hub, you have some breakdown and disruption, but you still have a significant network left to allow the cancer process to continue going on. But in the middle, with that intermodular block, you totally disrupt the entire network, and that target or that set of targets is going to give us the best chance of breaking down the tumor, because we're basically destroying the information. It's kind of like, cutting down a lot of telephone lines between, key cities which have the most access between them. Or, if you want to knock out the efficiency of flying from New York to California on American, you shut down, DFW Airport, which is a major hub connector. You would disrupt traffic much more than if you if you shut down an airport in Des Moines, Iowa, where there's very little traffic going through it. It may have some local effect, but it's not going to be a major disruption. Next slide. So, given all that, I now want to get into a specific example of what we've just been through, taking the information we get when we take the tumors from our patients and we send them off for analyses, and then how we can use that to try to figure out, how can we use this data to specifically manipulate the tumor with the therapeutics we have at hand in order to choose those patients with tumors that would most likely respond to the therapeutics we have. And we've recently, looked at an agent, MLN4924, which is an inhibitor of a process called Neddylation. It inhibits something called Ned8. We took patients who were going on that trial. It's already been initiated, and we sent that off, mainly to Foundation in order to give us, a readout as to what were the mutations and, what were the amplifications present. And even though it wasn't a large number of patients, what was very interesting is that the patients who were shown to have loss of a gene called CDKN2a, which basically is the gene which produces, as you'll see in a moment, a cyclin-dependent kinase, and yet had the wild-type p53 gene, 2 of those patients had a response to the therapeutic. Now, patients who had a loss of that same CDKN2a gene but had a mutant p53, those 2 patients didn't respond, and we looked at 13 other signals. Only 1 out of the 13 patients responded. So even though this is very limited data, it was enough for us to kind of come up with a hypothesis that, is there any way that we can find, that we can come up with a hypothesis, an explanation of why 2 patients with CDKN2a loss, but that still retain the normal p53 gene function, why this would actually, why could we explain this? And if we could explain it, maybe this is a group we could look for this particular agent. So the next slide. So this just gives you, where these genes are. It's on chromosome 9, and it's part of a complex that, codes, 2 important proteins, P16 and P14-ORF, and how it codes them depends upon what transcription factor is turned on or turned off, and whether or not exon 1B is the primary exon that is turned on, or exon 1 alpha is turned on, because depending upon which exon is initially turned on, 1 beta or 1 alpha, will depend upon whether or not you get a P14 or you get a P16. And as you can see, as you look down the slide, they actually function a little bit differently. The P14 affects something called MDM2, which has a direct effect on P53. It basically ubiquinates it and targets it, for proteasomal breakdown, whereas the P16 has an effect on other cyclins and on cell cycle. So they both kind of interact between the growth phase and the synthesis phase. In other words, the phase in the cell cycle, the cancer cell cycle in particular, since that's what we're talking about, where you're making the basic building blocks for DNA, and you're moving on to actually synthesizing or taking those building blocks and putting it together to make the DNA. So this just gives you an overview of what that CDKN2a gene, why it, how it functions, to give us some idea of, its potential importance and how we can explain how that might relate to P53 when it's mutated. Next slide. So how often does this inactivation occur? Well, this just gives you, an idea that this is actually seen, inactivating the CDKN2a is actually seen fairly frequently in a large number of cancers. We don't really have as good, gene data in ovarian, but we do have the mRNA and protein data, showing that even in ovarian cancer, the CDKN2a, gene is inactivated in enough to make this not only of interest in a reasonable number of patients, but to, potentially make it, if we can target this, something that could be cost effective because of the percentage of patients that show this inactivation. Next. So there are different ways in which the CDKN2a can be altered. It can just, there can just be an important deletion which renders the gene inactive. We can have a loss of heterozygosity, which means one of the two alleles, the maternal and the paternal allele, one of them is essentially non-functioning, so you just needed, some, spontaneous event happening on the other allele. I mentioned to you previously that you can have non-structural changes within the epigenome, and that can lead to a complete loss. And then you can have something called a point mutation, which can be a partial loss of the CDKN2a. So again, not important as to how it's altered, but the fact that there are many ways in which it can be altered. Next slide. So we know that this Ned8, which is basically what we, the therapeutic that we have is targeting. We're going to now look at Ned8 and how it affects MDM2, and then we're going to look at that relationship to the P53. So when you Neddylate MDM2, what you can see on the bottom, there are really three curves there. On the left, there are two curves that almost overlap, and that basically is the MDM2 levels. When you are using a mock therapeutic which doesn't affect Ned8, or essentially when you're using an ineffective Ned8. It basically tells you that in that case, MDM2 has a normal half-life of about a little over a half an hour. In other words, in a little over a half an hour, whatever the concentration is in the cell, it decreases by 50% every half hour. So in about a half hour, it's 50% of its original level. In 1 hour, it's 25%. In an hour and a half, it's 12.5%. So that's how we measure half-life. But when you actually look at what happens, with the Ned8 process, that actually stabilizes the expression of the gene, because when Ned8 is active, essentially what happens is that you are increasing the half-life by about 3 times. So you have a much higher expression of MDM2. Now this is important, and let's go to the next slide, because MDM2 can interact with P53. And it can interact with it in different ways, but however it interacts with a P53, it essentially, either by nedylating it, by going through one ubiquitin combination protein called 12, or it can ubiquinate it by going through ubiquitin conjugating protein 5, you wind up having a potential breakdown of wild-type P53 and rendering it inactive. So now we're beginning to hook up Ned8 with MDM2 and P53. So let's go to the next slide. So essentially, if you look at the middle, where you see that orange thing that says MLN4924, which is the therapeutic, and look at the bottom below the bifunctional arrows where we had just Ned8. The Ned8, when it's not inhibited, Neddylates, and we call this process of Ned8 modifying MDM2 Neddylation. It stabilizes the MDM2, and by stabilizing it, the MDM2 then ubiquinates P53, which results in its breakdown. Now, P53, remember, is mutated in at least 50% of all the cancers we see. This is a gene that is responsible for repair, for senescence. It's a gatekeeper gene. It is one of the most important genes that has ever been shown to be associated with the cancer process. So if we look at our ability to use a therapeutic to block NED, we can then rather than stabilizing MDM2, we can ubiquinate MDM2 rather than ubiquinating P53, break down MDM2, and then by doing that, we no longer have the MDM2 available to destabilize the P53. That results then potentially in a stabilization of the P53 protein, and since that is important for apoptosis or programmed cell death, that actually can lead to cancer cell kill. So by blocking NED8, we can affect the interaction between MDM2 and P53, but we can affect it in a way that, from just looking at this slide, should only work if you have P53 wild type. If it's already mutated and non-functional, then this whole rationale really should not be effective. And that kind of goes back to what we were initially seeing in those few patients. Next slide. So what happens then when we have the combination of a deleted cyclin-dependent kinase N2a and a wild-type P53, where we know that the CDKN2a inhibits MDM2. So when you essentially get rid of, when it's deleted, the CDKN2a, you have an increase in MDM2, and by increasing MDM2, you actually decrease the P53 protein, because MDM2, when it combines with P53, ubiquinates it and essentially results in its breakdown. When NED8 is present, NED8 essentially increases MDM2, and it also has a separate effect on P53, for which MDM2 has a feedback role. So when NED8 is present, you also wind up with a decrease in effective wild-type P53. So what we came up with was that if you knock out NED8 with MLN4924, you destabilize the MDM2. Now, when you destabilize the MDM2 by knocking out NED8, and you also have the CDKN2a deficiency, you basically then should have a significant increase in P53. And if that's the case, then we should have a much higher probability of programmed tumor death, a process that we call apoptosis. So this is taking the information that we found by deconstructing, by looking at the genomic changes within these patients that went on this protocol, and looking at those small groups that responded or didn't respond, to be able to use either limited data to see if we could find a rationale, and then allow that rationale to allow us to come up with a hypothesis that we could then present to the company and say, listen, rather than treating 100 patients with MLN4924, where you might have a 20% response or a 25% response, what happens if you try to target a population that has P53 wild type but has a deletion for CDKN2a? You might then have the chance of having a 50, a 60, or a 70% response rate in this particular tumor, which will enhance the probability that you'll get approval for your drug. And